Hello everyone and welcome back. This week in lab we are going to be examining fixed action patterns and how they relate to agonistic interactions between individual animals. So let's start first by defining some of these terms. All right, we're going to start with those fixed action patterns. We call them FAPs for short, as you can see right here. Okay. Now, fixed action patterns have a very specific definition. They have to meet two different criteria. The first is that these behaviors have to be stereotyped. All right? So, just like stereotypes for humans, stereotype behaviors are very consistent and highly repeatable, meaning the behavior is always the same, down to its tiniest detail. Now, these behaviors can be surprisingly complex, but none of them are more than a few steps long. Okay? And you're going to see examples of what this stereotypy looks like. So first, fixed action patterns are stereotyped. Second, these behaviors also have to be ritualized. It means that every single individual of that species, every individual of that kind of animal, performs these behaviors the exact same way. What that means is that the behaviors are universally recognizable to any member of that species. If every individual is born knowing how to do these behaviors, it means that they're going to be able to recognize this set of behaviors in others of their species, which makes it a good way to communicate. Now, these behaviors also have a couple other features that are important. They are involuntary, meaning animals don't actually choose when to engage in a fixed action pattern. These behaviors are genetically programmed. They're born knowing how to do them, and these behaviors have a trigger. Something that's seen in the environment, some stimulus that triggers the fixed action pattern in the animal, and once it's begun, they can't stop it. All right, so let's build on this knowledge a little bit more. And let's talk a bit more about that stimulus that I mentioned up here. Oops. Okay, so we're going to connect these ideas. All right. Now, the stimulus that triggers a fixed action pattern set of behaviors has to be very specific, and it also tends to be something very simple. Like several common stimuli that trigger fixed action patterns can be a certain shape combined with a certain color, like red circular spots on the body of an animal, angular shapes located on the body of the animal. Think like pigmentation, color patterns, that sort of thing. It can also be non-visual. It can be something like a specific scent, an odor that triggers the behavior. Or you can have a combination of two different visual stimuli. For example, a shape that's in motion. Perhaps something circular that rolls down a hill and then stops at the bottom. These are very, very simple stimuli, but they are specific enough that they will only trigger a fixed action pattern under appropriate circumstances. Okay? Now, we give these stimuli a special name. Stimuli that trigger fixed action patterns are called releasers. Releasers. Now, in lab today, you are going to have a chance to examine some fixed action patterns that are agonistic in nature, meaning they are based in conflict. Your study animal for this lab are these lovely individuals right here. This is a kind of fish called a betta. That's their common name. Their scientific name, though, is a little bit more of a mouthful. They're called beta splendens. Beta splendens. Right? This is a really interesting species of fish, and they lend themselves easily to the study of fixed action patterns and agonistic behaviors because they tend to be kind of grouchy, right? And they can get very, very aggressive towards other betas in their environment. Now, this is a sexually dimorphic species, which is very, very important. Sexually dimorphic. Di means two, okay? And morphic means appearance or how you look. Sexually dimorphic species have two different physical appearances for each sex. It just means the males look different than the females do. Probably not surprising then to know that we've got two different sets of pictures here. One set will be for a male, the other is for the females. Now, in the case of betas, the males, which is this handsome boy over here, tend to be very vibrant and flashy in color. And that's the biggest difference between the males and females, is these bright colors that only appear on the males. Now, the reason for that is this is an indicator to females of the quality of that male. If you think about the physiology of color, pigments, the actual molecules that make an animal colorful, are kind of hard to manufacture in the body. They take a lot of energy, they take a lot of raw materials and resources. So an animal like this guy who has lots of different colors, like the blues, the yellows, the purples, and the reds, he's having to get enough food to give him the energy to build all of those different pigments. And in addition to that, he's got to be able to collect enough raw materials from the environment in his food to keep those pigments fresh and bright looking. That's going to take a lot of effort on his part. It's not going to be easy to do. If he's strong, healthy, fast, and good at finding food, he'll be able to maintain his bright colors. If he's kind of slow, if he's not great at finding food, maybe he's not great at fighting for resources, he's going to be more drab in color. Of those two different males, the females are going to want the fast, agile, good hunter. They're going to want the male who is better at finding food to be the father of their offspring, which means they are going to selectively mate only with the brightest colored males, because those are the males that are probably the strongest. For that reason, we say that bright colors are a reliable indicator of mate quality. Reliable because there's no way that male can be that brightly colored without being good at finding food and without being fairly strong. Now, there are some physical traits that you're going to need to know about male betas in particular. Okay? You've got to be able to recognize where the mouth is. That's not too hard. Right? It's about where you'd expect it to be, right in there. Now, just back from the mouth and the eye in this area is a little flap called the operculum. Okay? Opercula is the plural for it. The operculum is a bony covering over the gills, but what you need to know about it is that these fish can swing this bony cover open, revealing the gills, which are back in here. Okay? They can use those bony coverings to signal other males by moving them back and forth, kind of like flapping. They basically flap them at another male. Okay? You'll see examples of that today. Now, in addition to the operculum, you've got to be able to recognize the pectoral fins. They're where you would expect the pectoral muscles on a mammal to be, right near what you might think of as the fish's chest. Now, just back from that, near where the butt is, all right, the anus is there. There's a fin that comes just before the anus, right here. It's called the anal fin. You also sometimes see it referred to as the ventral fin. Now, on the opposite side of the fish, on the top up here, you've got this fin, which anchors to the top of the body, called the dorsal fin, just like in a dolphin or a shark. And then finally, back here, this really, really big fin that makes up the tail, that's called the caudal fin. Right? Females have all of these different fin types as well, but notice how different they are in shape and color. The caudal fin and the anal fin are much smaller, as is the dorsal fin, and they're very drab. There's a pectoral fin here too, it's just tiny, it's right there. And these females also have the operculum, but they're not going to use it the same way as the males. Okay. So females have smaller fins, and they're drab colors. Drab means more natural. Okay? Now, the advantage there is these females are going to have a much easier time hiding from predators because they blend into the background. That's another reason why bright colors are a reliable indicator of mate quality, because this male is going to stand out no matter what he does. He can't hide. So if he's going to survive, he's going to have to be real good at avoiding predators. Okay. Now the reason the male has such big fins is to display those beautiful colors so he can show them off. Right? Females don't have to display to attract a male mate. Females are the ones who choose mates in this species. The males just have to wait to be chosen. Let's talk a little bit about the adaptations that these betas have for living in their environment. Those are going to be kind of key to understanding the behaviors, the beta behaviors that you are going to be watching today. So betas are native to tropical Southeast Asia, where they tend to live in very shallow, very slow-moving water, or even water that is stagnant. Stagnant water doesn't flow at all. It just sits there and gets kind of filled with algae and kind of muddy and gummy. Right? What all these environments have in common, all these different water pools where these betas live, is that there's very little oxygen available in the water. Now, this is going to be problematic for fish that only get their oxygen through their gills from the surrounding water. But betas have a bit of an advantage here. They're able to breathe both water, as you'd expect, but also air. You might see, if you have a pet beta, that they occasionally come to the surface of their tank and take gulps of air. It's because they can pull oxygen out of the air just as easily as they can pull oxygen out of water. But the adults aren't the only ones who need access to oxygen from the air. Both eggs and fry, which are baby fish, they also need access to oxygen from the air. There just isn't enough oxygen in the surrounding water. For this reason, males have a very interesting parental behavior. Males do what's called bubble nesting, where they literally create a nest of bubbles like you might expect. Here's how this works. Females will release eggs into the male's territory. The male, meanwhile, will release sperm in the same location that the female releases the eggs. So her egg and his little sperm cells will combine in the water, creating fertilized eggs. Now the male then does something interesting. He uses his mouth and he blows bubbles. So these are literally bubbles full of air. Okay? Just like a normal bubble would be. But then he takes these eggs and he moves them up and into these air bubbles. Now this serves two purposes. The first is that the eggs are in a little air-filled environment, but even more importantly, they're floating near the surface, where oxygen can diffuse from the open air down to where the egg is. Also, when the eggs hatch into little fry, like this little guy here, that little baby fish has easy access to the surface where all the oxygen is. So this is a species where the males engage in what's called parental care. They help take care of the offspring, which not all males in all animal species do. Now, males have a few other interesting characteristics, and these are actually the behaviors that we are going to spend the most time on in this lab. Okay? They're called agonistic behaviors, and they are almost always a competition between two males who are trying to impress a female. Either right then and there, they're fighting for her attention in the moment, or they are fighting to maintain control over a resource that they think will impress that female later on. Now, in betas, beta agonistic behaviors are all about territory, defending territory. And what's interesting is that the agonistic behavior in betas is a fixed action pattern. So it's a highly predictable, highly stereotyped, and ritualized behavior. Now, as you start to watch betas as they engage in these agonistic interactions, you'll realize that the releaser for this fixed action pattern has a lot to do with the physical appearance of another male. Specifically, it tends to be their color that's triggering. Color seems to be the primary releaser. Okay? There are a few different agonistic behaviors that males will take part in when they are presented with a rival or a competitor male. Okay? These are all fixed action patterns, every single one. Okay? Now, the first fixed action pattern is called a flare. Okay? This is where the beta, the male, will face his opponent and he'll open his opercula, those little bony coverings over the gills on either side of the face. I'm going to draw a really terrible diagram right now. Okay? So imagine this is the front of the fish. There's some eyes. There's fish lips. Okay? They will spread their opercula out to the side, so they'll swing them open, and it kind of makes their face look larger. Right? Now, the next fixed action pattern is the male will turn his side towards his opponent and do what's called a fin flick. That's the name of the next fixed action pattern. This is where they flick their fins. Okay? They'll open and flick a fin, usually a pectoral fin, in the direction of the rival male. Sometimes they'll flick several fins, like the dorsal plus the caudal fins. Or, I'm sorry, dorsal, pectoral, and caudal fins. All at the same time to make a big display. The next fixed action pattern is an escalation. This is where things start getting more heated in this interaction. It's called a charge, and it's exactly what you'd think. It's when the male beta swims straight at his opponent. He charges him. And if things go really, really badly in this interaction and the males are full-on fighting, the final fixed action pattern is a bite. This is where they will try to rip off each other's fins. Right? This is why you can't keep two male betas together in the same tank. Their colors will literally trigger each other's fixed action patterns over and over again. And because the males can't get away from each other in your tank, their interactions will always escalate to the bite phase every time, and they'll rip each other to pieces. Well, now that you can hopefully recognize the basic fixed action patterns present in betas, let's go ahead and talk about how you're going to use this knowledge in our lab today. Right? You are going to be using information from your lab manual. The chapter that you will need is written in the module. I'm going to show you that in a second. There are several activities in that lab that you're going to complete. Activity number 1 looks at just territoriality in betas. You are going to be measuring how often fixed action patterns occur and what forms they take in males that are battling over territory. There is a video in the module that you'll use for data collection, and it's labeled as such. Okay? There are 2 beta activities at the beginning of the video, just skip those and focus on the beta and mirror activity. There's a section where they give a beta a mirror. That's the one that you are going to use. Betas recognize the image in a mirror, but they don't recognize that it's them. They think it's another beta, and so they react aggressively. Now what's interesting is the more aggressive they act, the more aggressive the beta in the mirror gets, right? Because they're seeing themselves. They actually start to escalate their aggressive interactions towards their own reflection because they think it's escalating as well. Okay. All right, here are your methods. Okay. In the experiment, in the lab, okay, you assume that the number of aggressive displays a beta will give when alone is 0. If there's no releaser stimulus, there's no view of another male beta, the fixed action pattern for aggression won't be triggered, so there shouldn't be any aggressive displays. Okay? These displays will only happen when another male is present. Okay? Now, question 2 in your lab manual. Under the methods, you're just going to skip it. Okay? Now, in part 3, question 3, to collect the data for the experiment, and the experiment is a section in your lab manual. All right, so this is in the lab manual. For part 3 of this experiment, that's where you're going to watch the video and identify the aggressive behaviors you see for just the section where the beta is given a mirror. Right? Now, when you watch the video, you are going to pick 3 behaviors. Okay? It can be flaring. I would go with dorsal fin flicking specifically. Charging is another good one. There are others as well. But pick 3 behaviors, and what you're going to do is you'll count the number of times each of your 3 behaviors occurs per minute. This is continuous focal sampling. You are going to watch the video for all of its minutes, and it's not many, it's about 10 minutes long. Okay? And for each minute, you're going to record how many of each behavior you see. So this is an example data set. Right? Maybe in minute number 1, from 1 second to 60 seconds in the video, I see that beta do a single flare and 3 fin flicks, but no charges. Okay? Maybe in the second minute, so 61 seconds all the way to 120 seconds into the video, I see an additional 3 flares and an additional 5 fin flicks. So for each new box, don't include the data from the previous one, right? Just pretend these are all individual sets of data. So only record in each box how many times that behavior happened in that minute of the video. Right? And do that until the video ends. That's going to be your primary data set. There is one other activity from the manual that you will be completing. Okay? It's called the dear enemy effect. You're going to want to read the introduction to this in the manual, and then you're going to use a data set that I've already given you on Canvas to determine if the dear enemy hypothesis is actually accurate. Now, the dear enemy hypothesis, in short, is that males who already hold territory don't need to worry about other males who also hold territory. Okay, so here's the idea. Check out male number 1 over here on the left. This male has this nice chunk of the pond all to himself. Okay? He, though, shares one of his borders with male 2 up here. So this border they share. Now, male 2 could try to push into male 1's territory, but why would he? He's got a perfectly good territory himself, and all he would be doing was picking a fight with another male that he still has to live next to. Now, male 1 and male 2 may battle a little bit back and forth over this border, but it's not going to be a big war. Okay? Same thing with male 3. Male 3 has to share borders with male 2, male 4, male 5, and male number 1. So male 3 is surrounded by enemies. But all of those males also have their own territories. Right? They might squabble a little bit about where the boundary is between those territories, but those males aren't going to charge in and try to take male 3's territory. The primary reason why is because these males can only defend a territory of a certain size. Okay? If a territory is too big, they can't protect it. So once males have divided up a chunk of habitat, they really don't have to worry about each other. They're not friends, they're still enemies, but they're enemies that can tolerate one another because they know they're unlikely to challenge each other for territory. The idea of dear enemy is we use the word dear as an endearing term. It's like, oh, my dear. It's something we say about people we like. Dear enemy means that these males, even though they are enemies, have this somewhat cooperative relationship where they try to leave each other alone. The problem comes from males, like male A over here. Male A has no territory whatsoever, but he wants some. It's the only way he's going to get access to a female. So as male A cruises through all of these territories, he's going to challenge each of these other males for control of their territory. Male A is the only real threat to males 1 through 5. The males with territory aren't a threat to each other, but male A is. He will try to evict these other males from their environment. So, what sort of behaviors would you expect to see based on this dear enemy effect? Well, if you present, say, male 1 with male 3, what would you expect their interaction to be like? Would you expect it to be highly aggressive, lots of flaring and charging and biting? Or would you expect them to maybe be slightly aggressive towards each other, but mostly ignore one another? Well, since male 1 and male 3 share a border, if the dear enemy effect is real, you would expect their interaction to be mild, okay, or low-key, because they aren't threatened by one another. And the dear enemy effect also assumes that these males can recognize each other. Now, by contrast, if you present male number 1 with male A, our male who has no territory, this guy up here, okay. There he is. Okay. If you present male 1 with male A, now what kind of interaction would you expect? Well, male A isn't from around here. Male A is a complete stranger to male number 1. If the dear enemy effect is true, you would expect this to be a violent, aggressive interaction. This all assumes that the dear enemy hypothesis is accurate. If dear enemy is correct, this is what you'd expect to see. You will be presented with a data set in the module and instructions in your lab manual for Activity 2 that will allow you to evaluate that data and see if you think the dear enemy effect is supported by it. All right. Our next step is to go and take a look at all the resources that you have this week in the module. So here we are in this week's module. The lab that you're going to be completing is Chapter 12 in your Field and Laboratory Exercises Manual. Okay? This document talks you through the requirements for this week's activities. Okay? There's background information here, as well as where to find additional background information in the manual. Okay? There's also a little bit of terminology here that you should be thinking about. This will connect to our next unit in the lecture class as well, where we're going to talk about the scientific method. Okay. Now, there's going to be some questions in here that you will need to address. You are going to utilize the video entitled Agonistic Displays by Betas. Okay. Then you're going to use the worksheets in your lab manual to collect your data. And keep in mind that the male is looking at itself, so its own behaviors are going to cause it to react aggressively, right? Some of its behaviors are going to cause more intense reactions than others. This will help you get a sense of which display behavior is considered most threatening by betas. Okay? And then finally, you're going to test the deer-enemy hypothesis using worksheets in the manual and the data in a document I'm going to show you next. Now, there's a lot of questions in the worksheet this week. Feel free to write them out by hand, okay? And then just take pictures of the paper and turn that in. Now, you do have a lab write-up, okay? As always, follow the directions here. Make sure that you label the sections in your lab manuals. You're actually going to write out the word objective above your paragraph where you describe your objective. Write out the word procedures above the paragraph where you write out your procedures. Now we have a new section this week that we're going to introduce. Fittingly, it's the introduction. Okay? This is where you provide background information for your experiment and any animals that you used in your experiment. So for this one, this experiment, you're going to be briefly describing the biology of betas that we went over in this lecture and that's in your lab manual. Okay. All right, so if we navigate back to the module, here's the deer-enemy experimental data. Okay? It's actually just a PDF. So it shows you the territories for the males, okay? Gives you experimental protocols, and then the data that was collected during this experiment. So assume the experiment's already done. Okay? Now when you look at this territory, keep in mind that the males don't leave their sections of the pond. Male 1 only knows male 2. This is the only other male he ever sees. Male 2, however, also knows male 1. But in addition to male 1, he knows male 3 too. He knows who his neighbors are. Male 3 knows male 2 and male 4, but male 4 over here only knows male 3. Right? They know who they share borders with. Keep that in mind. Okay, so this is the data you'll use for the deer-enemy part of the lab. And then the video that you will need is here. Video titled Use for Data Collection. Okay? Now there are 3 ed puzzles this week as well. They're short videos and they give you more insight into how these betas move, how they adapt to their environment, and a little bit more information on their aggression. Okay, that's it for the week. Enjoy!